Now let's talk a little bit about genetic engineering. In genetic engineering, we alter an organism's genotype using recombinant DNA. We obtain the recombinant DNA as we described previously. We take a section of DNA and through molecular cloning, we insert that DNA into a vector. That foreign DNA then, through that vector, is put into a host. The organism that receives the recombinant DNA that has now been modified is referred to as a GMO, or a genetically modified organism. A transgenic organism is an organism that possesses DNA from another species. Whether that DNA is from the same species or another species, any sort of recombinant DNA put into an organism creates a GMO. If that DNA is from another organism, then it's a transgenic organism. Some common uses that we have for this technology is the development of antibiotics, vaccines, production of hormones, and modification of crop plants. Here we'll see several videos that discuss genetic engineering.
From the smallest single-celled organism to the largest creatures on Earth, every living thing is defined by its genes. The DNA contained in our genes acts like an instruction manual for our cells. 4 building blocks called bases are strung together in precise sequences, which tell the cell how to behave and form the basis for our every trait. But with recent advancements in gene editing tools, scientists can change an organism's fundamental features in record time. They can engineer drought-resistant crops and create apples that don't brown. They might even prevent the spread of infectious outbreaks and develop cures for genetic diseases. CRISPR is the fastest, easiest, and cheapest of the gene editing tools responsible for this new wave of science. But where did this medical marvel come from? How does it work? And what can it do? Surprisingly, CRISPR is actually a natural process that's long functioned as a bacterial immune system. Originally found defending single-cell bacteria and archaea against invading viruses, naturally occurring CRISPR uses 2 main components. The first are short snippets of repetitive DNA sequences called clustered regularly interspaced short palindromic repeats, or simply CRISPRs. The second are Cas, or CRISPR-associated proteins, which chop up DNA like molecular scissors. When a virus invades a bacterium, Cas proteins cut out a segment of the viral DNA to stitch into the bacterium's CRISPR region, capturing a chemical snapshot of the infection. Those viral codes are then copied into short pieces of RNA. This molecule plays many roles in our cells, but in the case of CRISPR, RNA binds to a special protein called Cas9. The resulting complexes act like scouts, latching onto free-floating genetic material and searching for a match to the virus. If the virus invades again, the scout complex recognizes it immediately, and Cas9 swiftly destroys the viral DNA. Lots of bacteria have this type of defense mechanism, but in 2012, scientists figured out how to hijack CRISPR to target not just viral DNA, but any DNA in almost any organism. With the right tools, this viral immune system becomes a precise gene editing tool, which can alter DNA and change specific genes almost as easily as fixing a typo. Here's how it works in the lab. Scientists design a guide RNA to match the gene they want to edit and attach it to Cas9. Like the viral RNA in the CRISPR immune system, the guide RNA directs Cas9 to the target gene, and the protein's molecular scissors snip the DNA. This is the key to CRISPR's power. Just by injecting Cas9 bound to a short piece of custom guide RNA, scientists can edit practically any gene in the genome. Once the DNA is cut, the cell will try to repair it. Typically, proteins called nucleases trim the broken ends and join them back together. But this type of repair process, called non-homologous end joining, is prone to mistakes and can lead to extra or missing bases. The resulting gene is often unusable and turned off. However, If scientists add a separate sequence of template DNA to their CRISPR cocktail, cellular proteins can perform a different DNA repair process called homology-directed repair. This template DNA is used as a blueprint to guide the rebuilding process, repairing a defective gene, or even inserting a completely new one. The ability to fix DNA errors means that CRISPR could potentially create new treatments for diseases linked to specific genetic errors like cystic fibrosis or sickle cell anemia. And since it's not limited to humans, the applications are almost endless. CRISPR could create plants that yield larger fruit, mosquitoes that can't transmit malaria, or even reprogram drug-resistant cancer cells. It's also a powerful tool for studying the genome, allowing scientists to watch what happens when genes are turned off or changed within an organism. CRISPR isn't perfect yet. It doesn't always make just the intended changes. And since it's difficult to predict the long-term implications of a CRISPR edit, this technology raises big ethical questions. It's up to us to decide the best course forward as CRISPR leaves single-celled organisms behind and heads into labs, farms, hospitals, and organisms around the world. Wrap your head around the implications of CRISPR with this playlist on genetics, or check out some of the other mind-blowing science being developed around the world and in the universe right now.
CRISPR. Its name kind of sounds like an ad for fresh vegetables, but it's changing the entire field of biology. Using this technique, scientists can edit genes with a precision, accuracy, and speed they never had before. And that's great news for research and human health. But if humans can now manipulate the genome of any organism, there are ethical questions that we need to consider. Any molecular biologist will tell you that genetic engineering is a tricky pain in the butt. The goal is to do one of a few things. You could change the amount of a gene in a cell or delete it completely. You can also make a mutation in a gene by altering, adding, or taking out pieces of DNA called base pairs. Or you can introduce a gene from a totally different organism, like how we made bacteria that synthesize insulin and how we use the gene for GFP, the glowing green protein from jellyfish, as a biomarker in lots of experiments. Traditionally, there are a handful of techniques that you can use to do these things, but none of them are really convenient. For example, you can use a solution with special chemicals or particles to get DNA into lab-grown cells, generally called transfection. These cells will express the gene for a few days, but you get way too much of the gene floating around and way too much expression. Plus, it isn't part of a chromosome with the other genes and regulatory bits, so you can't be sure it's behaving normally. You can also use a modified virus to deliver a gene, because they normally stick their genetic material into cells and integrate it with their host's DNA. So just kill off the make you sick parts, give it a gene you want to study, and let the virus do what it does best. However, this method has problems too. The virus doesn't care where it sticks the gene, so it could be smack in the middle of another one and cause a bunch of problems. And that's just individual cells. We also have these proteins called nucleases that can target and cut certain sequences of DNA, kind of like a tiny pair of scissors. One type, called zinc finger nucleases, can be customized to cut near specific sequences of bases in grooves in the DNA double helix. They've even been used to make a few human patients more resistant to HIV infection by breaking a gene that helps the virus enter our immune cells. But when you change the building blocks of a protein, it's really hard to predict what the 3D structure will be, so it takes a lot of time, and you have to engineer a brand new protein for each cut. Changing a whole organism, like knocking out a gene in a mouse, can take years, thousands of dollars, and generations of breeding, not to mention a bit of luck. Or it used to. Enter CRISPR, or more correctly, CRISPR-Cas9. CRISPR was originally described as a sort of immune system in archaea and bacteria. See, when some of these organisms get infected by a virus, they save a chunk of that virus's genome in this long charm bracelet-like string of DNA called a CRISPR locus. They use each DNA chunk as a template to synthesize a similar molecule called RNA, which does a lot of things in cells. Here, the RNA is a guide to find more matching DNA chunks. Cas9 is another one of these nuclease proteins, and it cuts wherever the guide RNA tells it to. So it's all about that RNA protein duo. The next time that virus shows up, the guide RNA finds matching DNA and calls over Cas9 to chop it up and destroy it. And here's the thing, a system where you can send one protein to cut anywhere in the genome just by giving it a chunk of RNA is really useful for molecular biologists. Making a specific sequence of RNA is much, much easier than making a specific 3D protein shape, like for those zinc finger nucleases. CRISPR lets you make a precise cut in DNA, so you can use it to knock out a gene or even trick the cell into inserting a gene. Just depends on how the cell fixes the broken DNA strands. Sometimes the cell just jams the ends of the DNA together as best it can. That method is prone to mistakes, so it's good if you want to break a gene. Other times the cell uses a template to repair DNA, and you can send in a template along with the RNA and Cas9, so it adds whatever you want, like a whole new gene. Since the template is guided to a specific part of the genome, you're going to have just the right amount of your gene integrated into a chromosome, unlike older techniques where you could get too many copies or even too few. So CRISPR makes gene editing faster, cheaper, and more accurate than ever before. Cue an explosion in genetic engineering research. CRISPR is mostly being used to edit genomes in cells at lab benches to learn how they function without a gene or with a new one. Some scientists figured out if you blunt the scissors of Cas9 so it can't cut, the protein will go where the guide RNA tells it to and just sit there, turning a gene off temporarily instead of deleting it. That way, Cas9 acts kind of like a light switch. Turn the gene off, see what happens, turn it on again, see what happens then. But like any technique, the CRISPR-Cas9 system isn't perfect. Sometimes it cuts in the wrong place. It might look at a sequence that almost matches the guide RNA and go, yeah, that's probably right. It can also insert sections of DNA the wrong way around, or delete sections. In lab-grown cells, this isn't a huge problem. You can just use the technique a couple of times and check for cells that got it right. And it's still way better than the inaccuracies from other methods. But doctors are also beginning to develop CRISPR as a gene therapy for patients, and making a wrong cut in the genome of an already sick human could be disastrous. If they have cancer, for instance, an extra damaged gene in the already mutated cells might make the disease worse. That's why scientists have been carefully engineering the guide RNA and the amino acid sequence of Cas9 to try to make the technique more accurate and cut the right sequence every time. They've made enough progress that human clinical trials involving CRISPR-Cas9 are making their way through the approval process, one at the University of Pennsylvania in the US and one at Sichuan University in China. Both are combining CRISPR with another cancer treatment called immunotherapy. Immunotherapy is based on the idea that a person's immune system can find and fight cancer with some help. Both trials will take some of the patient's immune cells and use CRISPR to give them a genetic cancer spotting boost. Then they'll put the immune cells back into the patient so they can get to work. One of the genes they're altering was also targeted by a drug in the cancer treatment of former US President Jimmy Carter, and researchers expect the CRISPR method to work even better. Editing an adult's genes in their own cells to treat them isn't that controversial, but CRISPR-Cas9 gives scientists the power to edit human embryos, and as you can imagine, this has caused quite a debate in the scientific community. The good news is, scientists are aware of the just because you can doesn't mean you should principle, and everyone is thinking very carefully about how to proceed. We've also thought about using CRISPR to kill all the mosquitoes, or at the very least alter their genomes, because some species transmit lots of deadly diseases. We do it by taking advantage of a naturally occurring genetic quirk in some insects called a gene drive. Since chromosomes come in pairs, most genes have a 50-50 chance of being passed on to offspring. Not gene drives, though. They code for a protein that cuts DNA, specifically to cut the other chromosome that doesn't have the gene drive. The cell goes, crap, how do I fix this? And the gene drive goes, look, chromosomes should match, so you should just copy me. So through the normal repair process, the cell ends up with a copy of the gene drive on both chromosomes, so it's definitely going to be passed down to any offspring. By doing this, gene drives can pretty much spread through the entire insect population after multiple generations, regardless of natural selection. And Cas9 can be engineered into a gene drive pretty easily. It already cuts DNA, so you just stick the gene for Cas9 in a mosquito chromosome, plus a sequence that'll become the RNA guide, and any extra template sequence. The RNA guide can target any mosquito gene you want, but only when the gene for Cas9 isn't present. So anytime that targeted gene is in the mosquito's DNA, the CRISPR-Cas9 gene drive will essentially cut and paste itself over it. In theory, we could target genes that affect fertility in mosquito species, like the ones that carry malaria. As the gene drive spreads over multiple generations, this would exterminate them forever. A less extreme idea would be to target genes that allow things like malaria parasites or the Zika virus to survive inside certain mosquitoes, so they can still bite people, but diseases aren't transmitted. By the way, if you're worried about it, this wouldn't really work in humans. Mosquito generations are short enough that a gene drive could spread in a few years. Human generations are not. You'd probably have to wait millennia to see any widespread effect. For the most part, CRISPR is revolutionary because it's a day-to-day tool for scientists. It's a way for them to learn more about their favorite gene and do more experiments. That's good for the public because it speeds up research and it's cheap. And we all want effective pharmaceutical drugs faster, like for cancer treatment. None of those experiments are going to escape and start a genetically modified zombie apocalypse because those petri dishes of cells are pretty hapless. They'll die if you leave them on the counter. Moving forward, research institutions and regulatory agencies are keeping a close eye on what CRISPR-Cas9 can do. Because this technique can do a lot of genetic engineering stuff, and it's accessible to a lot of people. And mostly, that's just really exciting.
Let's also briefly talk about the production of vaccines, antibiotics, and hormones using technology. The traditional way that we made vaccines was we would take a weakened form of the virus or an inactive form of the virus and inject that into an animal that would activate the initial immune response, and then also provide some memory to that antigen if the organism was exposed at a later time. More modern techniques can take genes of disease-causing organism, clone those into vectors, and produce an antigen. An antigen is typically a foreign protein that your body launches an immune response to, and you produce antibodies to attack that antigen. If we can just take the gene that makes the antigen that our body can recognize, clone that particular gene into a vector, then we can introduce this antigen that would stimulate our body to produce the immune response. The flu vaccine that we create every year uses genes that were cloned from the virus. One of our more recent techniques is to use messenger RNA that produces the proteins that our immune cells need to recognize the virus. When we create antibiotics, we use organisms like fungi that naturally produce antibodies to bacteria. When we synthesize hormones, we can use recombinant DNA to take a gene that would produce a particular hormone, such as insulin. We can put that into bacterial cells and get those bacterial cells to produce human insulin. One aspect of genetic engineering is gene targeting. Some of the classical methods for studying individual genes, which is what we're doing in gene targeting, is to start off with a phenotype and then ask what is the genetic basis of this particular phenotype. More modern techniques kind of go in reverse of this. They start with a DNA sequence and then ask, what does this section of DNA or this particular gene do? This is sometimes referred to as reverse genetics. In gene targeting, we take a recombinant DNA vector to alter gene expression. So we're very specifically trying to go in, find particular genes that are responsible for certain traits, and alter those traits. We can introduce a mutation in the gene that might cause a change in the gene, or it might delete part or all of that particular gene sequence. If I can alter a gene and then also notice what the phenotypic consequence is as a result of that, then I have some understanding of what that gene does. We've already looked a little bit at some genome editing technology that is fairly new and is revolutionizing genetic engineering, and that is CRISPR-Cas9. Here we'll look at another video that shows some applications of CRISPR-Cas9.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
We know that we can use genetic engineering both for diagnosis and therapy. Genetic diagnosis involves genetic tests that will determine an individual's likelihood of having a certain disease and allows us to learn a little bit more about that disease, even if it's already been diagnosed. Treatment and prevention plans can be generated based on these results. That then can lead to more effective therapies, in many cases very specific targeted therapies. So we're finding more and more as time goes on that rather than using a single drug to treat a particular disease, we're having more personalized medicine where the drug therapy is specific for a particular genotype. For example, doctors can identify traits and treatment options that are very specific to individuals and to that individual's disease. Not all cancers are caused by the same types of mutations, and so different treatments might be more appropriate depending on what type of mutation is causing that particular disease. In gene therapy, we use genetic engineering to cure or treat the disease. So we can use it for diagnostics and we can also use it for therapy. This image shows an adenovirus vector being used for therapy. We take a new gene that we want to inject into a cell. We insert it into the DNA. That modified DNA is now inside of a viral vector, in this case, which is adenovirus. That DNA gets injected into the cell, makes it into the nucleus, and now this new gene that we've added can be transcribed. So this good gene maybe can correct a mutation or a faulty gene that was present. We briefly mentioned before transgenic organisms. When do we have to go to using transgenic organisms in applying genetic engineering? Recall that there are many stages in protein synthesis in eukaryotic cells. Some of these eukaryotic mechanisms that we want to study, we can't really study in bacterial cells because they don't go through all the processing that a normal eukaryotic cell does. Remember, eukaryotic cells, after they make their messenger RNA, they add the cap and tail and they splice it, and then they have a lot of other proofreading mechanisms and other things that happen that we don't see in bacterial cells and we're not going to see in viruses. So we can use those organisms for some things, but there are certain types of treatments that we would really need a eukaryotic cell and perhaps an entire organism to really understand the full extent of this disease process. We can insert human genes into organisms like we've used sheep, goats, chicken, mice, creating transgenic organisms, and now we can actually understand a little bit more the physiological mechanisms behind some of those diseases that we couldn't really understand at the cellular level. We can get these other organisms to express human proteins. As long as the human genes are there, the mechanism for producing the correct protein is there. And so these other organisms can express these genes. We also use transgenic mice quite a bit to study the effects of mutation and recombinant genes. So transgenic mice have been used very extensively to understand disease process, particularly when we want to understand long-term effects over the life of an organism. Since mice have a shorter lifespan, we can understand those processes that occur throughout the life much more readily in a shorter-lived organism than we could in an organism that lives longer. We also use transgenic plants. Clearly, when we are looking at trying to genetically engineer crops for resistance to disease, resistance to pesticides, so we want to spray a pesticide on a plant, but we don't want to hurt the plant, so we want them to be able to tolerate that pesticide. We also can enhance their nutritional value, and we can make them have longer shelf life. A lot of different plants have been used incorporating genes for that effect. When we create any sort of transgenic organism, we have to be very careful because we have to watch what the potential effects could be on the natural species in an area. So if I create a transgenic plant, is that going to harm the natural ecosystem and the native species that are there? And we also have to be careful about the effects on human health. We want to make sure, for instance, that if I have developed a plant that produces a chemical that is a natural pesticide, that also isn't toxic or damaging to humans if we consume it. So we obviously have to be very careful with that. We've taken recombinant genes from bacteria, we've put it into plants, and that has allowed plants to have a natural insecticide. One of the first GMOs was the flavor saver tomato. This gene that was added slows the ripening process so the tomatoes don't get soft as fast, they don't rot as fast, they don't get fungal infections as readily, and it also increases their shelf life. It's no longer on the market because there were problems maintaining and shipping them. It gives us a little bit of an idea of some uses for genetic engineering.